11s, we're going to be looking at effective digestive systems. So this is slides 102 through 109. Starting at 101 though, which was our last slide from our last video, we've just got our 6 efficient or effective digestive system features here. I'm going to be referring back to those. I've got them on the board behind me, so don't worry too much if you don't remember them perfectly, but we're going to refer back to them. We're going to draw a few things up on the board. This is a skill you need to have yourself. So firstly, overall makeup of the mammalian digestive system is very consistent. For mammals, our gut, our digestive system follows this sequence. We have a mouth, esophagus, stomach, small intestine, a cecum, which is the early part of the large intestine, a large intestine, a rectum, and the anus. However, the size of these different organs varies and depends largely on diet. So for example, herbivores need a far longer gut and much slower gut than a carnivore. Now, if you think about the quality of the food that we're getting as meat eaters, the reason that our gut needs to be faster is because the likelihood for things having grown on that food that perhaps aren't particularly good for us. So you think like chicken, we have risk of salmonella. Meat, we have the same sort of risks. So, or meat in general, I should say, have the same sort of risks. So our gut needs to be faster, so it reduces the amount of time the bacteria actually have where they could actually set up a colony in our gut. Whereas herbivores, the quality of their food is that it's normally quite clean, but the concern is that that has very little nutrient value. Particularly, the majority of the sugars, majority of the carbohydrates are bound up in the cellulose, the cell wall of the plant. That's very hard to digest. If you try to eat bark, you'll find that out pretty quickly. Because we're omnivores, we're in the middle. So we eat both plants and we eat meat. So therefore, we have a stomach or a gut that tends to be quite slow compared to carnivores, but certainly much faster than herbivores. So to put it into context, it takes us roughly 24 hours to work through our food, whereas a cow, which is a herbivore, will take up to 72. So there's a big difference there. So our first thing is, well, what's our first feature? We're looking at the mouth and the esophagus. So primary role of the mouth is to taste food and accept or reject it. So this comes into this factor of capture and handling of food. How are we going to handle this food? Is it safe to eat? So our mouth features here. Now the bit that you need to be adding in is the additional stuff. So the fact that what's the mouth doing? Well, it's capturing the food obviously, and it's also handling it. Do we accept it? Do we reject it? The teeth then begin physical digestion. So this is our physical breakdown. It also starts chemical digestion. So last video we talked about this salivary amylase, which is able to break down the polysaccharide starch into maltose, which is a disaccharide, meaning 2 simple sugar molecules, 2 monomers, if you remember that term from last lesson. The pH of the mouth is between 6 to 8. So it's between slightly acid to slightly basic, depends on what you've eaten. And this is the ideal range for amylase to work through. Once we've chewed the food, it goes into the esophagus, which moves towards the stomach. Now let's focus a little bit on esophagus. What have we got here? Well, the esophagus typically will only allow things to go one way. It's got a sphincter at the end of it called the gastroesophageal sphincter, which prevents the stomach acid from coming up. So we have this one-way breakup. So the esophagus fits into here. Oops. Physical breakdown, not so much in the esophagus. Capture and handling, not so much. So we've really got the esophagus here, our one-way gut, but there's also efficient transport. It's moving from the mouth down into the stomach. So the esophagus sits here as well. We're not concerned about high temperature, there's no exchange taking place now. So the food is swallowed, enters the esophagus, and at that point the food is called a bolus. Now, the key feature of a bolus is that it will still look like the food that you have eaten. So, because then it gets stuck, you cough it up, it's called a bolus, you cough it up, and it still looks like the chunk of apple that you haven't chewed properly because you were too hungry to do so. So the esophagus moves the bolus via this wave of contractions called peristalsis. I kind of imagine it, it's like if somebody were to have a sausage casing that they filled with mince, and they're trying to push the sausage out because there's the meat out because there's something in there that they're not really interested in. You squeeze at the top, then you keep squeezing, and you squeeze further and further and further down until it comes down at the other end, which in our case is the stomach. The esophagus is attached to the stomach, and it's separated from the stomach via this gastroesophageal sphincter. Now, a sphincter is just a ring of muscle. It's a ring of muscle, and in this case, it opens when it receives a signal to say there's something on the esophageal side, but it will snap shut as soon as that is gone. So this wave of contractions pushes the food to the sphincter. The sphincter receives that there is something there. It relaxes, the food is able to drop into the stomach, and then it snaps shut again so that the acid isn't able to come up. That's why in some cases with babies, they have a malformed or they have an incomplete gastroesophageal sphincter, and this causes reflux, which is that constant cycle back up. There's potentially other reasons to do with problems with the digestion of milk and that sort of thing, but quite often it can just be that they have an immature gastroesophageal sphincter. So here we have our first features. We have our mouth capture and handling, physical breakdown. Esophagus helps with the one-way gut. It's also transport and storage. Moving on to our next set of organs. We're moving quite quickly because the concepts here are quite basic. It's memory stuff that we've got. So it's the definitions, the connection between the affected features and these different organs is what's critical. So the stomach itself acts as a storage organ. So here we have our first bit, transport and storage. So the stomach works there too. So it's involved in storage, and it says chemical and physical digestion. So here we have our physical breakdown. The glandular tissue, so remember glandular tissue is that specialized epithelial tissue, that specialized barrier tissue that secretes certain chemicals. In this case, glandular tissue produces hydrochloric acid and secretes that. It produces a protein or a pre-protein called pepsinogen. Now, pepsinogen is broken down by hydrochloric acid into pepsin, which is a protease. Now, here is the clever thing about that, and these are the sorts of questions you should be asking yourself. Why? Why would the body make pepsinogen when it could just make pepsin? It could do that. Surely evolution, the hundreds and thousands or millions of years that evolution has gone on for, surely we could have sorted that out. What evolution has worked out is that pepsin is I suppose you'd say it's not selective, or it's not particular to only our food. It will break down all proteins, which means that the proteins that make up our stomach will also get broken down, which is incredibly unhelpful. If you release pepsin into your stomach and it starts to break down your own stomach, that's counterproductive. So it releases it as pepsinogen, pushes it out, and there's this special little lining, a mucus lining that protects the stomach wall, and the pepsin can't get through that. So once it gets into the hydrochloric acid into the stomach juices, the pepsinogen is converted into pepsin, and it's able to do its job. The hydrochloric acid means that the stomach is incredibly acidic, a pH of between 1 and 3. Now this is perfect for all of the proteins that are in the stomach, but it also denatures, that is a fancy word, denatures. It means permanently removes function or permanently changes shape. In this case with a protein, if you change the shape, it doesn't do what it's supposed to do anymore. So the salivary amylase loses its shape, can no longer break starch down into maltose because of the acidic conditions in the stomach. It's also protective in terms of infection. A very acidic environment is very difficult for bacteria to survive in, or viruses to survive in. The epithelium, the wall, the lining of the stomach is protected from this incredibly acidic environment by mucus, which is produced by glandular tissue. Very little absorption of nutrients occurs in the stomach. In fact, the only absorption that occurs in the stomach would start occurring in the mouth. Chemicals that we eat that are able to dissolve directly across the cell membrane. Now, particularly here, we're talking about alcohols. Alcohol starts to actually act in our bodies, or starts to be absorbed by our bodies as soon as it enters our mouth, because it's so effective at passing across the cell membrane. So any absorption that happens in the stomach has already started in the mouth, and even potentially in the esophagus. Alright, so here we have our features. Physical breakdown, stomach, transport and storage, stomach. Not worried about high surface area because we're not worried about exchange yet, but now we are. First major exchange organ. long and has incredibly high surface area. So here we go. Small intestine there. Just going to shorten intestine to INT. The high surface area of the small intestine is achieved by millions of these tiny microvilli. Now, if I might change, I might use red here. So the small intestine is covered by these folds, and these folds are called villi. And on top of these, and sorry for those watching at home, it's a very small, but they are called microvilli. Incredibly small. I might draw that slightly larger. I might like that slightly larger. So effectively what you've got is you've got folding of the small intestine, and then on top of the folds you have further folds. So the small intestine itself is folded. And then the folds of the small intestine themselves are folded. So we have this repeat increase in surface area, and you immediately think when you see that, why? Think about the lungs. Why do we have the alveoli? Well, here, what have we got? In the lungs we had exchange. In the small intestine we've got exchange, but what are we exchanging? Well, we're looking for the sugars, the carbohydrates. Hopefully at this point they've been broken down far enough that we can digest, or that we can absorb them directly. If not, the small intestine also has proteins that it produces. We've also got fats, we've got nucleic acids, we've got proteins, so amino acids, we've got the fatty acids, we've got the nucleotides, we've got vitamins and minerals. It needs to all get in. So these microvilli on the epithelium of the small intestine help that happen. The epithelium is also very thin. Typically it's only one cell thick. So between the epithelium, between the food and the blood vessels, the capillaries, and the lymphatic vessels, there's normally only one cell. This is incredibly helpful because it means that exchange can occur more rapidly. Remember, diffusion, thickness of the membrane is what's going to be driving it. So the things that enter the blood supply, and this is a little hint for you, a little reminder here for you. Things that enter the blood supply: carbohydrates, amino acids, nucleotides. Fats, fatty acids and glycerols go into the capillaries, the lymphatic capillaries, or the lymphatic lacteals. So every villus, every fold has its own capillary network, based of blood capillaries and based of lymphatic lacteals, or lymphatic capillaries. These are just specialized lymphatic capillaries. So the small intestine has roles in moving of digestion as well. So the chyme moves from, now when food goes from the esophagus into the stomach, it's called chyme. When it goes from the stomach into the small intestine, the name stays the same. So the chyme moves from the stomach into the first section of the small intestine, which is called the duodenum, and it moves along again via peristalsis. So these waves of contractions of squeezing shut and opening, and squeezing shut and opening, so that the food can move in one direction. So a one-way gut, here we have it again. The small intestine helps with that. And we should actually add in that the stomach does the same. Helps with our one-way gut and with compartmentalization. So we can really, by the end of this, you'll see that every single organ of this body, of this system actually works to help with that. The chemicals that are released, so and pancreatic enzymes, so that comes from the pancreas. Bile, which is emulsifier, so it helps to split up fat. From the liver, enzymes from the small intestine itself. These chemicals, these enzymes, this bile, complete digestion of carbohydrates to monosaccharides, proteins to amino acids, lipids to fatty acids, and glycerol. So here we have again our physical breakdown. Specifically, if we're thinking only physical digestion, we're thinking of bile. I'll just fit that in here. So our small intestine, S-M-I-N-T for that. The bile itself is alkaline, so the chemical that helps to split up physically digest the fats is alkaline. So it raises the pH of the chyme from 1 to 3 in the stomach to 6 to 7. And this allows these new enzymes to function while also breaking down while also denaturing the enzymes from the stomach. Here we've got our diagram of the small intestine. I'd strongly recommend you spend some time looking at that, making some notes on it, but we're not going to do that now. Moving on to a little bit more detail. So the proteins in the cell membranes. So if we're thinking proteins in the cell membranes, we're thinking this is either facilitated diffusion or perhaps even active transport. They transport nutrients at very high speed into the capillaries, into the lacteals that are a part of each villus. This consumes considerable energy. If it's consuming energy, it must be active transport. So it consumes considerable energy, and the body itself needs to make sure that this is actually working efficiently. It needs to make sure that it has enough energy. The question there is how, and we're going to have a short discussion around that in class this week. The fatty acids, the glycerol, travel across the intestinal cell membrane, very easily because they are dissoluble in fats and then reassemble into fats in the lacteals. The lacteals themselves are a part of the lymphatic system. They are and so they're a part of that, so they're under very low pressure. The second part of the small intestine, the ileum, so this is a secondary part of the small intestine, continuous absorption, but with very little digestion. pH here continues to rise from 6 to 7 to 7 to 8, and this slows the intestinal enzymes, but it doesn't stop them entirely. Now the other thing, we've talked so much so far about nutrients. Water is also a considerable part of our food, certainly a considerable part of the kind. 90 to 95% of the water that enters the small intestine is absorbed. So by the time our food leaves the small intestine and enters the large intestine, we have quite a solid, not very liquid material anymore. The blood leaving the intestine enters the liver directly via a vein called the hepatic portal vein, and this is where nutrients may be stored before moving them on to the rest of the body. So the liver is this processing site. Everything that goes into our bodies has to go to the liver first. It's like if you turn on a Pura tap, the filter is what the water runs through before you can actually drink.